Okay, hi everyone. Let me see if I can get the light better. Sorry, it's kind of dark here. Okay, so this is our first live stream review. Feel free to ask whatever questions you have, and I can see your questions, and then I'll ask them. I'll just answer for everybody who's watching right now. The one person. All good. Hopefully, we have more people join us. Okay, so to review, your quiz covers pages 32 to 47. There was a question today, so just to clarify, it 47, the bottom half has like eukaryotic gene control. You don't need to know that. So just up to basically prokaryotic control, I think, right? Let's see, you should definitely be able to transcribe and translate a sequence on the practice quiz. There are several examples of that. Well, the practice exam and the practice quiz, and then also make sure you can do one of those transformation problems, ones we did in class, the one that has like the all summer and then tables. But go ahead and ask whatever questions you like, and go from there. Anyone? Things you're not sure about? I would definitely say take a look at the practice quiz and the practice exam. Remember that you do not need to know the codon table and the promoters. Like, I will give you both of those things. You just want to know what to do with them, right? Okay. I will keep talking until somebody asks a question, so feel free to fire in anytime. I hope I can actually see what you're saying. Let's see. Yep, okay, I think we're good. Okay, so you know what? I think one of the things I'm going to do right now is walk you through how to do the transcription and translation problems, because I find that we've gone over it, but people generally have trouble with it even so. So let's kind of talk for a minute about how you would do that. If you want to have the practice quiz open, that might be helpful. Just because, like I said, there are questions like that. In fact, I will pull it up, so I will walk you. Well, actually, I take it back. I want you to do that question on your own. So I'm just going to walk you through it conceptually, and that way, hopefully, the instructions give you an idea how to do it. When you have a, oh, hello. Yes. Okay, so Isha, let's talk about 5' and 3'. That's perfect in the context of what I'm about to talk about. So remember that DNA doesn't really have like a left and right side, but instead you have the 5' and 3' ends, right? So essentially, if you go back to your notes, it has to do with the orientation of the carbons relative to the sugar and the phosphate. So if you take the sugar, right, and you have the oxygen at the top, and you start to the right of the oxygen, and you go clockwise, it's carbon 1, 2, 3, 4, and then 5 is like the chimney. And so carbon 3' and 3', 3' and 5' just sort of refers to if you're looking at the end of the molecule, is it closer to carbon 3 or closer to carbon 5? In the end, what this means is you have like directionality to your DNA. So you always build DNA or RNA 5 to 3, meaning you always hook it onto the 3' end. So if I'm reading from like the earliest piece I put onto the latest piece, it would go from 5 to 3, right? And so it just gives us a sort of a way to figure out direction on the DNA and RNA strands. So let's talk about it in the context of synthesis. Let's say I have a piece of DNA, right? I'm always going to give you both strands. So one strand, it's all relative, right? So one strand will be running 5 to 3 in the direction of transcription. One will be running 3 to 5 in the direction of transcription. You want to be careful that it doesn't have to be like left to right or right to left, right? Because it really just depends on where you're finding the clues. So what you're going to look for first is your promoter sequences. If for a prokaryote, that's minus 10 and minus 35. For a eukaryote, that's minus 25 and minus 75. But the idea is that they are relative to each other. So, sorry, sometimes I grab random visual aids to help me here. So let's see. Let's see what I can find here. Oy, sorry. Okay. I'll grab my children's toys to show you. All right. So let's say we have a strand of DNA, right? And these two are my promoters, right? Ghost is minus 10, and then doggy is minus 35. In this case, since doggy's on this side, minus 35 is on this side, and minus 10 is on this side, then that tells me that the direction that RNA polymerase will travel when it actually transcribes has to go towards the +1, right? It's going to go that way. Okay. Whereas conversely, if I had a different piece of DNA and minus 35 was on this side and minus 10 was on this side, now the RNA polymerase would travel this way. Okay. So the way that you can kind of figure out which direction RNA polymerase is going to go is you look for those clues. You figure out, so yes, Tanisha, it's going to tell you, it doesn't tell you which one is minus 10, which one's minus 35. That is dependent on the sequence. So minus 10 and minus 35 have different sequences. So think of them as like different spellings, right? So minus 10 is always TAT-AAT. You don't need to memorize that. I'll give that to you. Minus 35 is, I think, TTG-ACA, right? So what you do is you look for those sequences and you look for them the right distance apart. So my advice is always look for minus 10 or minus 25 because they're the ones that are easier to find. Minus 10 and minus 35 are actually the same sequence. They're just in different locations because they're different organisms. But so yes, so Lakshmi, the promoters are always going to be minus 10 and minus 35, or it might be minus 10, minus 25, minus 75. You have to look for the gene and figure out, am I talking about a eukaryote or prokaryote? So if you're looking at a eukaryote gene, it's going to be minus 20, minus 25, and minus 75 you look at. If it is a prokaryote, then you're going to look for minus 10 and minus 35. For the sake of simplicity right now, let me just say it's a prokaryote. Okay. So let's say it's a bacterial gene. So first I would look for minus 10, TAT-AAT, put a box around it, right? Now, in order for it to actually be minus 10, that means if I go 25 bases in front of it, right, earlier, I should find minus 35. So if I count backwards about 25 bases and I don't find my minus 35 region, TTGACA, then I know, okay, this is actually not minus 10. It's something that has the same sequence as minus 10, but it's not minus 10. If it is minus 10, you're going to find that sequence, TTGACA, which is minus 35, 25 in front, right? Or if it's a eukaryote, you'll find minus 25. And then if you count 50 in front, then you would find minus 75. Okay. So let's say I find those sequences, minus 10, minus 35. Now I can draw an arrow. I can essentially say, well, if I know where minus 35 is and I wouldn't know where minus 10 is, I can draw an arrow. Is it going this way or this way, right? In terms of where minus 25, minus 35 are located. Once I have that arrow, what it tells me is what direction RNA polymerase will travel in. And that's where 5-3 becomes important too, which I'll get to in a second. Technically, the promoter is going to look for, the RNA polymerase is going to look for those promoters on both strands, even though the strand I give you, like the sequence I give you, actually is always going 5-3. But technically, just know that RNA polymerase is actually looking for that double-stranded region. When it finds that double-stranded region, then the next thing it's going to do is it has to then start transcribing a short ways after that. So for our purposes, what you would do on those problems is you would say, okay, let me take minus 10 or minus 25. But again, let me just stick with minus 10 for now. And you're going to pick the middle T or the middle A. And you're going to say, okay, one of those is the actual minus 10 nucleotide, meaning it is 10 in front of the +1, right? And so if I pick that minus 10, then I'm going to say, okay, this is my actual minus 10 base. I'm going to count forward. Minus 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, +1. Remember, there's no 0. And wherever I land on for +1, circle that. That is your actual beginning RNA nucleotide. Now, here's where also that 5' 3' comes into play, right? Because you know what direction RNA polymerase is traveling. And so now you have to look at, for given that direction, which of the two strands is going 5 to 3, and which of the strands is going 3 to 5. The strand that is going 3 to 5 in the direction of transcription is your template strand. That's the one that RNA polymerase would physically look at and build its complement. The one that's going 5 to 3 in the direction of transcription is my coding strand, which technically doesn't actually participate in transcription, but remember, as a shortcut, is actually going to tell me the sequence of my RNA. So I find my +1. That's where I start transcribing from. You can either do it the shortcut way, or you can, just actually be the RNA polymerase and look at the template strand and put in its complement. Okay, so that's how you transcribe. Then when you translate, what you're going to do is you take your RNA now, or as a shortcut, you can look at the DNA, right? But you take your RNA and now you're looking for your start codon. Remember, the start of transcription is not the start of translation. Look for the AUG, and then only after that do you start counting off by 3s and match up the codons using the codon table. But hopefully that kind of gives you an idea of how to do those problems. Okay, so going to questions. I'm actually going to go a little out of order just because Lakshmi was asking about something that has to do with this, and I'll answer Lakshmi's question, and then I'll come back to Chaleen's question. So if there's a mutation in some of the nucleotides in between the promoters are removed, but both promoters are there, would this mean? Yeah, so it would actually, which is, it's one of those things where, remember, in order for transcription to happen, you have to have the promoters with the right sequence, so the right spelling, but also the right spacing. So if you were to delete a bunch of nucleotides between the promoters, even though the promoters are still there, you make them really short, like the distance between them becomes way shorter. And if they're way shorter, it basically means RNA polymerase, which has those two recognition, has essentially like two binding regions, won't find them because they're not spaced correctly apart. And so if the spelling is wrong on the promoters, or the spacing is wrong, or both, then you would assume that RNA polymerase would not find the promoters and therefore would never start transcription. If you never start transcription, you would never start translation. Okay, Shalene's asking about enzymes and cleavage. So cleavage is essentially the idea that sometimes in order to activate an enzyme, you have hydrolytic or digestive enzymes that will cut off part of that enzyme. And in doing so, anytime you take off part of a protein, then you end up changing its shape. So in this case, that could now cause the protein to change its shape, and now before it wasn't working and now it is, or maybe before it was working or wasn't working and now it is, right? So a really good example where that happens is actually in your small intestine. You produce a inactive version of a protein digesting enzyme called trypsinogen, but then enzymes in your small intestine will come in and essentially through cleavage reactions break off part of that inactive enzyme. And when you break off that little piece, it restructures the shape of the enzyme and now makes it functional and starts actually digesting protein. Okay, anything else you guys have thought of as I'm talking a lot? Annie says, on the TAT. Okay, so remember you start at the middle T or the middle A. So what that actually means is on a quiz or test, when you count forward like 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, or for minus 25, minus 25, 24, all the way down to +1, right? There's actually two correct answers. So as long as you give me one of those two correct answers, you're good. I don't expect you to give me both. Just give me one of them, right? But either the middle T or the middle A. And remember, when you count forward, no 0. Yeah, so microRNAs, all I really need you to know about them is they are, first of all, very small pieces of single-stranded RNA. And at the same time, there's not too much we know about them, right? We're still finding out a lot about them. But what they've basically found out is these little pieces of RNA can be regulatory molecules. They think that one of the major things that happens is that the microRNA will basically bind to a piece of RNA, right? Like, let's say an RNA that's supposed to be translated. And the problem now is when you have this microRNA. Sorry, I'm looking for a visual aid again. Let me see if I can find something. It's whatever is immediately available here. So let's say you have a piece of RNA, right? And then a microRNA binds to it, right? By complementing it. By doing that, now it becomes a double-stranded region. So that's actually going to impair or stop translation, because when that happens, then the ribosome can't translate something that's double-stranded, right? So they think essentially that these microRNAs serve as a way to regulate possibly translation, like, within cells. Tanisha, can you explain how ribosomes move during translation process? I don't need you to know too much about how they move. I mean, I think you would think of it, just think of it as like they're inching down the mRNA like 3 at a time. Past that, I don't need you to know much more. What I would say you want to be sort of aware of is the idea of like what's happening right in the ribosome. So the idea that you have the ribosome is closed around the mRNA, and then you have all these tRNAs flying in, and they're basically trial and error. They're trying to see, do they match up with the mRNA? If they match, they stay. If they don't, they leave, right? And in the process, they're moving through those 3 sites: the A site, the P site, the E site. The A site is like the lobby where it comes in to see, does it match? And then if it matches, it stays. And then from there, it moves into the P site, where essentially the enzymes on the ribosome will take that amino acid off of the tRNA and add it onto the growing chain. And then it goes to the E site, where it exits, and then it leaves. What does rRNA do in the translation process? Essentially, for our purposes, rRNA is part of the ribosome, right? So to construct a ribosome, it's a combination of protein and rRNA. And so they together allow that ribosome to catalyze the reaction of taking those amino acids off of the tRNAs and putting them onto the growing chain. But it's not like our RNA does like one specific thing. In fact, they're still trying to kind of figure out, because I think for the longest time they thought that the proteins had to be the ones that were like enzymatic in the ribosome and whatnot. But now they're finding, actually RNA might have some of those enzymatic qualities as well. So for our purposes, our sake, just know is that it's part of the ribosome and therefore participates in translation. Let's see. Okay. Let's see, what else can I talk about? Do you understand how operons work? Oh, yes. And he says, can I describe the difference between nonsense and missense? So number one, I actually don't need you to know the names. What I mean by that is, sure, tell me the names, that's okay. But more specifically, I would want you to be able to look at any sequence and figure out what is the impact of said mutation, right? So nonsense is an example of where either in some way, shape, or form, you basically introduce an early stop codon. It could be that things are deleted and now you have a stop codon that shows up earlier than supposed to, or it could be that you've added a bunch of stuff and in so doing you added a stop codon, right? But that's a nonsense, is when essentially the protein becomes too short because it can't finish translation because an early stop codon is introduced. A missense mutation is really just the idea that you have a change to your amino acids. That can be extensive. It could be like everything after that is screwed up, or it could be like one single point mutation where like one nucleotide changes and then the whole thing, everything's off after that. So you could have like one missense or extensive missense. Cassidy is asking, can I talk about operons and how they work? Yes, definitely. So operons, remember, only happen in eukaryotes, not us, okay? And essentially they're groups of related genes. So the idea is that if instead of like individually transcribing and translating all the different genes and proteins that might be needed for a particular process, you transcribe them all at once. There's one promoter, RNA polymerase hops on, transcribes the whole thing, usually 3 or 4 genes, and then it will basically having that one long piece of RNA be able to make all the proteins from that. So we have our repressible operon and our inducible operons. A repressible operon means that it's normally on, but it can be shut off. A inducible operon means it's usually off, but you can switch it on. In both cases, what you have is you have another region that's totally not part of the operon itself, which is our regulatory gene, and that gets transcribed and translated pretty much like no matter what, right? Regardless of whether that operon is on or off, the regulatory gene is being transcribed and translated. So you have a regulatory protein being produced. The big question in this case is that particular protein functional? Like, meaning, is it going to do anything or not? So if we go with something like a repressible operon, that means it's normally on, but you can switch it off, meaning normally the repressor protein is in an inactive state. But when if I basically bind, in this case, the molecule, right, then I can activate it, and now it will start to repress or stop transcription. Whereas an inducible operon means that it's normally off, but you can switch it on. So normally the repressor protein already goes and binds and stops that, but it can then switch it on when needed. There's also the positive gene regulation for the operons, right? That's the idea of like it's already on or off. But then when it does that, it will suddenly now stop working. Okay, there's a question about the poly A tail. So let's talk about that for a second. With the poly A tail, that's part, remember, only in eukaryotes, not in us. So in that case, the idea is the poly A tail is part of the RNA processing that happens between transcription and translation. It gets added on, you can assume, by enzymes in the nucleus, where you add basically a whole bunch, like 100 to 300 A nucleotides onto the end of the RNA. And remember, again, it's not the same as like the termination sequence. And it's thought that it does two things. So number one, it helps to sort of point the RNA out of the nucleus, but then also makes the RNA more stable, meaning like you now have this like huge string of A's where you can have enzymes like degrade it and it's not going to mess anything up. So it kind of gives you a little more buffer room, right, for that all to happen. So I'd say when is just between transcription and translation. And again, what it does is it helps sort of direct things out of the nucleus. Okay, in eukaryotes, does mRNA go to the rough ER or free floating? So that depends, right? It does both, actually. So normally what happens is ribosomes make proteins no matter what. But if you have a protein that is meant to be exported or excreted, right, then that's going to be a ribosome that attaches to the rough ER. Remember, that's a whole process where you have a signal peptide. So now as a signal peptide, after it's translated out, then the SRP will bind to it, drag it over to the rough ER, attach it and stuff it in, right? So if it's a protein that's supposed to leave the cell, then you would see the mRNA being made on a ribosome attached to the rough ER. But if it's a protein that's going to stay in the cell and not go anywhere, then it would be free-floating ribosome in the cytoplasm. In the end, they're still going to make protein in the same way. It's just a question of, after it's done, is it floating around the cytoplasm or is it stuffing it into the rough ER? Okay, what else have you guys got? Anything? Let me see, what else can I talk to you about? No problem. Let's see. Trying to think of common mistakes that I see. We already talked about the transcription translation. You do not need to know specifics of the lab, although I don't think they're actually in that page of the range of pages I gave you anyways. Let's see. You definitely want to, I would say mutations are a big part of it. I mean, you can see it in the practice problems, right? But you want to be able to tell me in, not necessarily with names, but in terms of process, like how the mutation would impact production of said protein. Whether it's like you make no protein, or it's just different, or it's too short, or all those different things. Let's see. I think in terms of specific changes that happen to pre-mRNA, I would want you to just know those 3 steps. Know that you add the 5' G cap, you add the poly A tail, and then you remove the introns and put all the exons together. Those are the 3 things I would want you to know for, pre-mRNA to mRNA. Let's see. Any other questions? Let's see. Let's see. I'm going to look at the pro. Oh, a mutagen, Mitchell, is just anything that causes a mutation, basically. So it's a fancy name for something that causes a mutation, right? It could be like UV light is a mutagen. Yeah, so again, don't worry about the 47 part that has to do with gene expression 1 in eukaryotes. My intention was for you to basically cut it off right before then. Yes, Annie, I can totally talk about positive gene regulation. So positive gene regulation is the idea that you want to, instead of just like deciding if the operon, is going on or off, that you are actually now, once it's on, that you have the ability to like turn up the volume. So that's where you get cap and camp, right? Specifically, we look at it with the lac operon, the idea that assuming you already have lactose and the operon is on, then you sometimes want to make it go faster. So that's why I always call it like a cheerleader, right? Where you already had floating around in the cell, essentially, what could I call it? The cap, right? Cap is already there. It's not doing anything, but it's there. And if you assume now that you have, if you assume now that you've got the camp that's now present because you don't have a lot of glucose, then the camp goes and binds to the cap. It basically makes RNA polymerase more likely to bind to that location, and so therefore it speeds up transcription. Let's see. Can you about what you have to know about what? Yeah. So again, all I want you to know about changes is I would want you to know the 3 major steps. Add the 5' G cap, add the poly A tail, and then take out the introns and put together all the exons. That's what I would want you to know. Let's see. Anything else you guys think of? Let's see. Sorry, lots of messy stuff behind me. Let me think. Let me look at your notes or something and see what else might be in there. Okay. Is the particular... No, actually the promoters are universally pretty much the same. I mean, there can be small adjustments, but you can assume that for any given eukaryotic or prokaryotic promoter, that it would always be those sequences. It makes it easier for the RNA polymerase to recognize, right, than having to recognize a lot of different things. So that's why I will give you the promoters. Like, you'll just have to look for them. No, again, you don't need to know the sequence. On the day of the quiz, I will actually give you both a codon table and I will write out for you those promoter sequences. So you just know what to look for and how to find them, but you don't need to memorize them. Let's see. Don't forget your references on the website. Yes, I'll remove that. So again, counting for each promoter sequence means I would look for -10 or -25. And that's always going to be, in both cases, TAT. So I would pick the middle T or the middle A to count from as my actual -25 or actual -10. So if you're counting for -10, you'd say -10, 9, 8, 7, 6, 5, 4, 3, 2, 1, +1. And if you're doing -25, it would be longer, -25, 24, et cetera, all the way down. The main differences between prokaryotic and eukaryotic protein synthesis, the ribosomes are technically a little bit different, but procedurally they're about the same. So the main differences are going to be, number one, the RNA processing. Remember, prokaryotes don't do it. Prokaryotes basically do transcription and translation. That's it. Nothing in between. Whereas for a eukaryotic, you're going to do transcription and then RNA processing and then translation. And then other differences also have to do with then location. So the prokaryote can basically do everything at the same time, even as it's still finishing transcription, it can start translation on that piece, right? Because it's all in one room. Whereas for eukaryotes, it actually has to do it in two separate steps, where in the nucleus, it does transcription, and then it's going to move out to the ribosome, or sorry, out to the cytoplasm, where then it will do translation. Okay, what else? And again, don't memorize the sequences. We'll tell you them. Yes, feedback inhibition, Annie, is just the idea that usually you don't have like one enzyme working all by itself, right? Typically what you have instead is this whole chain reaction of stuff that happens, but rather than trying to block it at every step of that like whole chain reaction, the idea behind feedback inhibition is, what if sometimes the end product actually goes back and serves as a molecule to say like, hey, there's enough of me, you can stop now. So that's feedback inhibition, because it's like the end of the line is giving feedback to the beginning and saying, hey, there's enough of me, you can stop now. Yeah, we can go over the all, some, or none tables. So let me see. Let me think of what the best way to do it would be. You have the ones from class. If you don't have them anymore, you can go to the website and re-download it and try it again. For those, remember, those are essentially like, sorry, other noises from this side. Essentially, the all, some, or none tables are basically an application of operons, right? The idea that you have this bacteria that's trying to get a plasmid, and the plasmid, like I said before, is like a happy meal. So you get all the different stuff, all the genes in the operon. And so you have to sort of figure out then, like, based upon the descriptors, what is going to be true. Usually you have to figure out, first of all, like who's growing and who's not growing. Lab-wise, you don't need to study any particular lab. You want to understand just the content from 32 to 47. So with the table, right, the idea is like based upon the genes in that plasmid that will determine what you get. And so I would go row by row. Each time you're just sort of determining first, like who's growing, right? Transformed, untransformed, or both. And then you answer questions about that, where it's like, okay, of the ones that are growing. So if it's transformed and untransformed, how, which ones of those are going to have blank, blank, and then it could be all of them, some of them, or none of them. Or if it's only the transformed ones, then you would say, okay, of the transformed ones. So I think I already mentioned it, but you don't need to know specific labs, and you just know the concepts behind the labs. Elongation from the RNA attaching to the strand up to, yeah, pretty much. Elongation is the extension or building of the RNA then. Let's see. Other questions that have come up? Let me think, what else could I tell you guys? A signal peptide. Okay. A signal peptide has to do with a protein that needs to be exported out of the cell. So one of the ways to direct it to the rough ER is, well, the only way to do it is with a signal peptide. So it's actually the very beginning of said RNA. It has a sequence for what we call signal peptide, which is the first couple string of amino acids. Then those will be recognized by the SRP, which is a signal recognition particle. When it does that, it temporarily stops transcription, drags it over to the rough ER, binds to a receptor in the rough ER, and now it's going to start feeding that protein into the rough ER as it's being made. So the signal peptide is really that sticker, tag that says, like, take me to the rough ER. Okay, hopefully that answers questions there. Trying to think what else. Let me think. Let me see. I'll pull up our lab, our notes. I don't have them in front of me, and see if there's anything that comes to mind that maybe you should know. Unless you guys have more questions, in which case I'll just answer those. Like two. Okay. So, with enzymes, make sure you know what enzymes do, right? How they catalyze reactions that, so pages 32 and 33. Let me see if more questions have popped up. Nope. Okay. So if I go through, the different types of ways you can recognize the enzymes, including optimum temperatures and pHs, those descriptions. Make sure you know the difference between the template strand and the coding strand. Sorry. Entergonic versus exergonic. That has to do basically with the idea is, if you look at the reaction, is it absorbing energy or is it releasing energy, right? A reaction that you have to put energy in or is absorbing it, we would consider endergonic, whereas exergonic means it's releasing energy. We usually talk about that in the context of like reactions and things like that. Okay, I'll look again at our notes, but like I said, feel free to put any questions in there. Yeah, we talked about all the processes of transcription, translation. Make sure you know, like I said, you don't need to know the stop codons or know the idea of how transcription and translation stop and how those are different. Make sure you talk about mutations and what they might or might not mean. Let's see what else. haven't seen anything else pop up in the question feed, so I'll keep talking about random stuff. Unless you guys have other stuff to talk about. Yeah, we did mutations, operands. British dictionary, I think almost the end. The blue painting in the background. Oh, thank you. Mr. Chow took that picture. They like your picture, Mr. Chow. They like the San Francisco picture. Oh. No, you do not need to know how to calculate transformation efficiency. That is not on this test. You need to understand the general idea of transformation, for the all, some, or none tables, but that's about it. Let's see, what else? Let me look at our notes here and see if I can think of anything else. Yeah, I think I already mentioned before, you do not need to know specific labs, especially since it stops at 47. Let's see. I would say practice. If you haven't yet tried the practice quiz, make sure you practice it by doing the quiz. And what I mean by that is don't do it with your notes in front of you. Try it like a real quiz and then come back. And if you don't understand something, study those sections, come ask me questions, etc. I'll be around tomorrow, brunch and lunch. No tutorial. You can always email me if you have questions tomorrow too, or come see me. Lunch, brunch, etc. I am not sure what else to tell you guys right now. I think we've kind of covered everything. Let's see. You do, yeah. I think all the other stuff's pretty self-explanatory in terms of numbers. You don't need to know restriction enzymes or anything like that yet. Any last lingering questions? Yeah, I would know really generally what the sites on the ribosome do, right? Like the A, P, and E, where A is where they come in, it's like the lobby. P is where it's going to take the amino acid off the tRNA, attach it to your growing protein chain, and then E side is where it exits so that it can now go find another amino acid that is the same as it. Yes, the two DNA chains are attracted to each other by hydrogen bonds, not covalent bonds. Covalent bonds are what hold a single strand together, but then the hydrogen bonds are what make the two pieces come together. So those are fairly easily broken, when you need to do something like transcription. Okay, looks like we're almost at the end here. So any last questions before I go? And if you think of something tomorrow, like I said, just come and see me. It's not a problem. You're welcome. I'm glad it was helpful. I hope you guys get some rest. And you're welcome. 7th period, I'll see you guys tomorrow. Remember that if you didn't finish the problem set, please come and finish it because I have to cut it off then so I can post answers. Yeah, and just come see me if you have questions, okay? You guys are very welcome. Have a good night. Okay. Bye.